Close enough. Okay, well, I just want to thank everybody for coming to Colloquium. And this is it's a new format for Colloquium this year. The genetics department agreed that we'd use our fall for a sort of inward focus. And so we'll be hearing a lot of talks from our own trainers and other people on campus in the fall, as well as, and this is the really new part, as well as our fifth year students and some of the postdocs from the department. So this is a great opportunity for us to see the latest breaking science out of our program. And thanks for coming and being a part of it. So I'll let Valentina introduce our first speaker.
Okay. Hello, everyone. For those of you that don't know me, I'm Valentina Losardo. I'm an assistant professor in the Cell and Regenerative Biology department. And today is my pleasure to introduce my wonderful student, Elsa. She's just starting her fifth year, right? Yeah. So it's been 5 years with me. So she's the bravest of all of you. And she's been the first student also of my lab. And today she will present a story that we recently actually published in one of the AHA journal. And we think we discovered something really interesting about understanding genetic predisposition to cardiovascular disease. So Elsa, take it away.
All right, thank you both for lovely introductions. And yeah, I'm excited to share, so we'll get right into it. So I look into heart disease along with the rest of my lab. So the focus of our lab in general is studying genetic variants linked to complex human disease. And there's a few challenges that are associated with this type of work. A lot of these variants reside in non-coding portions of the genome, and they're poorly conserved. A lot of them are also not necessarily causal. They increase your risk of a disease, but they don't necessarily single-handedly cause it. So you're not necessarily looking for a very clear kind of disease phenotype when you're looking for what they do. In addition to this, these variants are often in close proximity, and they can be in very, very strong linkage disequilibrium, which causes them to inherit together. And if they inherit together all as one piece, it can be difficult to pin down which of the variants within a given region is actually the one that matters for what you're looking at. And of course, finally, both the genetics of the human body and the biology of disease are all going to have tissue and cell type specific effects. So if you're not looking at the right model, the right tissue, the right cell, you might completely miss the relevant piece of what you're looking for. We look specifically at heart disease, at coronary artery disease. It is the most common kind of heart disease. And most of us have heard of this disease. It's characterized by the formation of atherosclerotic plaques that block the coronary arteries, and they can break off and cause a thrombosis somewhere else in the body. And like I said, a lot of us have heard about it, and most of us probably think about it in connection with lifestyle factors like, poor diet, sitting with the posture of a cooked shrimp all day, every day, which is certainly something I do. But there's also a very significant genetic component. This is a disease that's about 40 to 60% heritable, and around 300 loci in the human genome have been linked to risk of CAD. Of those loci, around a quarter are predicted to be linked to the vascular wall, whereas only 10% are predicted to be lipid related. So that really points to the cells of the vasculature as important players in the progression of the disease. But despite this, the roles of various cell types within the progression of coronary artery disease remain incompletely resolved. So we look specifically at vascular smooth muscle cells, or VSMCs. VSMCs comprise the majority of the wall of a human artery, and they're known to play multiple central roles in the progression of coronary artery disease. So your average atherosclerotic lesion is going to have around 70% cells that were once smooth muscle cells. In addition to the contributions they make to the plaques, they're also known to undergo all kinds of really weird phenotypic switching during the progression of CAD. They can transdifferentiate into smooth muscle or into macrophages or foam cells, and those cells so closely resemble like normal macrophages and foam cells that they were actually indistinguishable for a long time. It wasn't until someone did a lineage tracing study that we found out that a bunch of these immune cells in the atherosclerotic plaques were actually smooth muscle cells that had transdifferentiated. Aside from that, they can take on any one of an array of modulated phenotypes where they can take on features of other cell types, but they don't completely transdifferentiate. And examples of those include things like fibromyocytes, SCM, or fibrochondrocytes. So we want to know, how do the CAD risk loci that have been, reported to increase risk of this disease affect the cells of the vasculature? Specifically, we focus on one locus, 9p21.3. This was the first locus in the human genome ever to be linked to risk of coronary artery disease, and it remains to this day the most impactful. It was initially identified in 2007 in a series of genome-wide association studies, and it was a pretty interesting region for them to have identified as having such an impact, because it's not only a non-coding region, it's actually a gene desert, so it doesn't contain any genes at all. Coding or non-coding. It is around 60 kilobases long, and what it does contain are around 80 single nucleotide polymorphisms that are in very, very strong linkage disequilibrium. And this linkage disequilibrium creates a haplotype that has two alleles: the risk allele at this region, which increases your risk of coronary artery disease by about 30% for every copy of the risk allele that you have, and the non-risk allele, which does not create this increased risk of CAD. It is worth noting that although the region does not contain any genes, it does overlap a long non-coding RNA called ANRL, which I just had appear on the diagram in blue. And you can see it does overlap the risk region by quite a bit, but there is a small portion of the risk region that does not overlap with the gene. This region is only conserved in higher order primates, which has pretty deeply affected prior research into it. So although it was identified close to 20 years ago now, we still don't have a really great idea of what exactly this region does or how it does it. And part of the limitations on that has been that we can't use animal models like mice. There have been some studies that have attempted this. So mice do have two genes, CDKN2A and 2B, which in humans are the two closest protein coding genes to the 9p21 region. But they don't have the 80 single nucleotide polymorphisms, which in humans define the risk allele. And they don't have the long non-coding RNA that overlaps the region. So there have been studies to try to knock out a region that is thought to be approximately orthogonal to the 9p21 region in humans based on its relative location to CDKN2A and 2B in mice. But this study didn't find any connection to atherosclerosis. So there's also been other work that has not attempted to utilize mice to study it, but these have been pretty limited to observational studies on medical cohorts or patients, which can, of course, uncover incredibly valuable correlative evidence, but it can't establish a cause-effect relationship between the genotype and any phenotype. So to get around this in our lab, we use human cells. So we use induced pluripotent stem cells, which we get by obtaining peripheral blood mononuclear cells from a donor with the genetic background of interest, and we somatically reprogram those cells to stem cells. Of course, once we have the stem cells, we can make any type of cell we want, like the VSMCs that we study. Other advantages of this system include maintaining the genetic background of interest while also erasing the epigenetic marks that would have come from the donor's lifestyle choices or previous medical interventions. Additionally, these cells can be cultured indefinitely, pretty much, whereas primary cells collected from actual human arteries, they senesce pretty fast. We can only passage them a few times before they stop dividing. And of course, with iPSCs, we can genetically edit them, which is the really crucial part to be able to design experiments that can prove a cause-effect relationship between a genotype and a phenotype. So we've used this system with a donor who is homozygous for the risk region at 9p21 and a donor who is homozygous non-risk. And we went through this process somatically reprogramming their cells to create induced pluripotent stem cells. Now, once we had those iPSCs, we genetically edited them to produce isogenic knockouts in both of those backgrounds. So that way it produced the 4 genotypes that we use: the risk cells with their corresponding knockouts that are genetically identical, except for the loss of the 9p21 risk region. And then, of course, the same in the non-risk background, the non-risk intact and the non-risk knockout. So once we had those genetically edited iPSCs, we differentiated them to our cell of interest, the vascular smooth muscle cells, and we started comparing them to see what kinds of differences were showing up in the presence of the risk haplotype. And pretty much right away, we saw a very, very strong transcriptional difference between the risk cells and all the other genotypes. So if you look at the top panel of the figure that just appeared on the slide, the red balls on that diagram, those are the risk cells. The two green groups are the non-risk background cells, and the yellow ones are the risk knockout. And you can see right away that the risk cells are off by themselves in the bottom right portion of that figure, very, very distinct from the others. And interestingly, the risk knockout cells are much closer to the green groups of cells, the non-risk cells, than they are to the risk cells. So what that tells us is that the deletion of the 9p21 region was sufficient to almost completely rescue the 3,000 plus transcriptomic gene difference that we saw being driven by 9p21. So that proves a cause-effect relationship between the risk region and this transcriptomic change. Additionally, we looked at the expression of this long non-coding RNA that overlaps the region, ANRL. And what we saw was that over the course of differentiation from stem cells to vascular smooth muscle cells, there was an increase in ANRL expression in our risk cells relative to our other genotypes. And when we overexpressed ANRL, we were able to partially reproduce the transcriptomic changes we'd seen driven by the risk haplotype. Specifically, ANRL was able to reproduce the decrement we saw in the top 6 downregulated genes in the risk cells relative to all the other genotypes. But interestingly, it wasn't able to reproduce the top 6 upregulated genes that we saw increased in the risk cells relative to the other genotypes. So we know it's playing a significant role in the 9p21.3 mechanism, but it might not account for the entire phenotype. So our question is, does 9p21 drive a vascular smooth muscle cell state resembling a previously reported CAD disease state? And if so, by what genetic mechanism does it act to produce this altered disease state? So this brings me to my work and a recent publication, which used the same model with the 4 genotypes that I described, the risk intact, the non-risk, and the non-risk knockout. But this time we sent them for single cell sequencing to dig a little deeper. So here I have the single cell data that we produced. The column on the left of the panels that just appeared, that's our single cell data, the iPSC-derived VSMCs. And the column on the right, you can see is labeled healthy human heart arteries. That data is from a publication that collected primary cells, so endogenous cells from a donor. And what we're doing in this figure is comparing the expression of these 3 genes, Tagalin, CNN1, and ACTA2, which are vascular smooth muscle cells. And we're looking at their expression in our iPSC-derived VSMCs and how it compares to the expression of those same markers in endogenous cells taken from an actual artery. And you can see the people on Zoom won't be able to see me pointing, but it's labeled on the UMAP for the healthy human heart arteries. The smooth muscle cell population is in that kind of like corn-shaped mass on the center left of the UMAP. And you can see if you compare how those markers are expressing in the endogenous smooth muscle cells versus how they're expressing in the iPSC-derived VSMCs, there's a strong similarity. So that tells us that our iPSC-derived cells are in fact reflective of something you would see in an endogenous human artery, and not just kind of a series of culture artifacts. But to further confirm this, we collaborated with Dr. Clint Miller's lab at the University of Virginia, and they have an atlas of human arteries, the Metaplaque Atlas, which is comprised of multiple single-cell data sets taken from both healthy and atherosclerotic human donors. So these are all endogenous cells. And we asked them to project our iPSC-derived VSMCs into their atlas. And sure enough, the smooth muscle cells from the atlas are in the center of this UMAP in pink, and our cells are overlaid in gray. And you can see that there's a substantial amount of overlap. Once again, confirming that this model is something that's useful to study human disease and is something that's going to be relevant potentially for clinical advancements. So having confirmed that, we then wanted to see what our genotypes were doing in this data set. So on the left here, I have our single cell data set again, colored by cluster, and on the right, I have it colored by genotype. And right away, you can see our two green groups, the non-risk background cells and the risk knockout cells in yellow. They overlap quite a bit. They're all intermixed in those three regions of the UMAP. But when we look at where the risk cells, they exhibit, once again, their unique transcriptomic profile. So once again, confirming that the deletion of the region essentially reverts the transcriptomic profile back to normal and that the risk have a very unique transcriptome. But we wanted to see where our clusters were grouping with respect to these genotype groups. So we made this bar chart and we identified clusters 3, 4, and 6 as containing the majority of our risk cells and only negligible amounts of the other genotypes. So we proceeded with those for analysis. So having identified these clusters, we wanted to see biologically what's driving them, what's different about them. So we did a gene ontology analysis. You can see the clusters, they're labeled on the left, cluster 6, 4, and 3, the risk clusters. And then we separated the genes by the upregulated and downregulated genes. And we saw a very interesting pattern emerge. So if you look over at the downregulated genes, some of the terms that are boxed include cell substrate adhesion or wound healing. These are terms that you would expect to see in happy, healthy VSMCs, but here they're being downregulated. And if you look over at the upregulated terms, you can see that there are terms related to cartilage development, connective tissue development, and a whole bunch of neuronal developmental terms in cluster 6. And those are obviously not something you would expect to see in vascular smooth muscle cells. So what this told us is that even though our risk cells are expressing smooth muscle cell markers, they're not behaving normally for vascular smooth muscle cells. To further confirm this, I did a trajectory analysis, which involves plugging our single cell data set into a computer program that assumes that there is a transition somewhere in this data, and I ask it to tell me what that transition is. So we anchored all our trajectories in cluster 2, which is the green cluster, and that has approximately even amounts of all of our genotypes. So it makes sense as kind of a central starting point. And then we did a separate trajectory analysis on each of our genotypes. And what we saw was that all the trajectories proceed from the left to the right across the UMAP, but the non-risk, the risk knockout, and the non-risk knockout trajectories, they all go down as they proceed from left to right across the UMAP. And they end up in that orange and purple cluster on the bottom right of the UMAP. The risk trajectory, on the other hand, it goes up as it proceeds from left to right across the UMAP, terminating in that turquoise cluster, cluster 4. So this confirmed for us that the risk cells are diverging from the other genotypes. Finally, we also looked at the transcription factors driving these trajectories, and we saw that there were 2 transcription factors that were common to the risk knockout and the non-risk knockout. Those are in orange. There were a few that were in green. Those are the ones that are common to non-risk, risk knockout, and non-risk knockout. So all of the genotypes aside from the risk cells. And then we had a good number of transcription factors marked here in red that were unique to the risk trajectory. So that told us that these trajectories are driven by different modules of transcription factors. Interestingly, we also saw a decrement in the risk cells in this gene, LGAUS3. This is a gene that's been reported in a variety of modulated VSMC states connected to CAD. And it's considered a marker of plasticity. So the fact that it's reduced in our risk cells, and specifically that it appears to be showing up less than in the healthy human heart artery data, where you can see that it does show up in a normal amount. This tells us that the risk cells are transitioning to whatever this aberrant state is, and then they're getting stuck and presumably losing the ability to respond normally to stressors or environmental stimuli. So having established that the risk cells take on this aberrant state and they appear to be stuck there, we wanted to see if we could match this aberrant cell state to any of the previously reported vascular smooth muscle cell disease states reported in CAD. So one of the first things we checked were these 3 genes, which are classic VSMC modulators. We know that they tend to change the phenotype of VSMCs during the progression of coronary artery disease. But you can see here that there weren't really any genotype-specific patterns. So whatever the risk state is in our data, it doesn't appear that any of these classic modulators are the ones responsible for driving it. We also wanted to check for transdifferentiation and transition to a few previously reported disease states. So here I have as examples macrophage and fibromyosite. And what we did here was we took a similar approach to what we did previously, comparing the expression of key markers for different cell types and how they express in our data versus how they express in the native cells where they're supposed to be. So you can see that kind of crescent-shaped group of cells in the top row of UMaps on this panel that just appeared. They're showing the expression of those two genes nice and bright. They're really, really purple. But those same genes are pretty much not present at all in our data on the bottom row. And we did this with a few more cell types and more markers for each cell type, but I'm showing some examples here. We also checked for markers of this state, the fibromyosite state, one of the modulated states. And you can see this gene TNFRSF11B. It is not super, super strong in the fibromyocytes, but it is very, very unique to them in the data set where they're supposed to be, the diseased atherosclerotic or the atherosclerotic human artery data set on the top row of this figure. But that gene is pretty much not expressed at all when you look at our data on the bottom row of this figure. So it doesn't look like we're seeing transdifferentiation to immune cells, and we're not seeing transition to this fibromyocyte state. What we did see was an enrichment for these genes, SOX9 and collagen 2. And these are genes that are known to be involved in osteochondrogenic development, so bone and cartilage cells. And when we dug a little deeper, we saw a concomitant decrease in collagen 1A1 and 1A2, which you can see on that violin plot. And when we looked at actual markers of osteoblasts and chondrocytes, sure enough, we saw that they were enriched in our risk cells relative to their isogenic knockouts. So this pointed really strongly to the idea that the risk haplotype is driving these cells to take on an osteochondrogenic-like state. Now, our collaborators in the Miller Lab at the University of Virginia, they saw a state like this in their Metaplaque atlas. So they did this analysis. The figures I'm showing you right now are all from their publication, where they took the smooth muscle cells from their atlas and they did a trajectory analysis similar to the one that I did on our data. And what they saw was that the smooth muscle cells appeared to start with the contractile smooth muscle cells, proceed through transitional smooth muscle cells, and then they could end up as either fibromyocytes or fibrochondrocytes. And as the name suggests, fibrochondrocytes are VSMCs that have been modulated to take on features of osteochondrogenic-like cells. So when we saw this, we asked them to project our cells into this transition to see if our risk cells were overlapping with the fibrochondrocytes or with any of the kind of precursor fibrochondrocyte states. So here is the initial integration of our iPSC-derived VSMCs with their data. And here it is again with the different cell groups annotated for subpopulations. So you can see our iPSC-derived VSMCs in gray. They're mostly overlapping with that peach-colored population in the center of the UMAP, and those are transitional smooth muscle cells. But here is the trajectory analysis that they did on the integrated data sets. And we see the same trajectory that they saw with contractile smooth muscle cells at the top, and the trajectory moves downward through transitional smooth muscle cells until it gets to fibromyocytes and fibrochondrocytes. However, we also have a population of cells that it's extending downward past where the fibrochondrocytes are. And when you look at the breakdown in that window in the top right of the figure of what cells are present in that region, you can see it's pretty much only cells from our data set, not from the atlas, and it's almost exclusively risk cells. So it looks like the risk cells are proceeding along a very similar trajectory to the fibrochondrocytes. They're also moving from top to bottom, but it looks like they're moving even further in that direction than the native fibrochondrocytes did. So what this told us is that our risk cells do bear a significant resemblance to the fibrochondrocytes that we see in actual human arteries during the progression of coronary artery disease. But we might actually be getting an even more extreme version of the phenotype than what we see in the Metaplaque atlas. So now that we've established this very compelling transcriptional signature for this osteochondrogenic state, we wanted to see if it was impacting the function of the cells at all. So one of the first things we looked at was calcification in these cells. Of course, you would expect bone and cartilage cells to have more calcium than other cells of the body. But this is also a very interesting phenotype for us to look at because the human arteries are known to calcify during coronary artery disease. So this is a clinically relevant phenotype. And we stained the cells for calcium using alizarin red, so the calcium should show up red in the staining. And we looked at the cells at baseline, but we also treated them with calcification media to see if they responded differently to culture conditions that encouraged calcification. And what we saw was that even at the baseline, the risk cells exhibited statistically significantly greater calcification than other genotypes. And after treatment with the calcification media, that difference became even stronger, which you can see quantified in the top right bar chart here, comparing the risk to their isogenic knockouts. We further confirmed this with 2 additional risk donors, as well as a non-risk and a non-risk knockout donor, which is the bar chart on the bottom right of the slide. And we found that this is something that remains consistent both across genotypes, but also across donors. It's not just something we're seeing in the cells from one person who maybe was a fluke. This connection between 9p21 and calcification is precedented in the literature. So it has been reported in connection with calcification in GWAS studies, and at least one study found that people who were homozygous risk for the 9p21 CAD risk region exhibited statistically significantly greater calcification in their atherosclerotic plaques than people who were homozygous not risk. Interestingly, that same publication also found a connection between the calcification and the stability of the plaques. So that's something that matters quite a bit for the prognosis of coronary artery disease. If you have stable plaques that are less likely to break, then you're less likely to get thrombosis as a result of it. So you want more stable plaques. And what we're seeing here is that apparently calcification is correlated with reduced stability of the plaques. So also something that's potentially very interesting and clinically relevant. We also looked at the migratory capabilities of these cells. Cells that have, or smooth muscle cells that have taken on osteochondrogenic-like features as a result of CAD are known to exhibit reduced migrative abilities. So we did an assay where we seed a layer of cells and we put a scratch down the center, and then we see how long it takes those cells to fill in the wound. Now the top row of figures here, those photos were taken the day that the scratch was made, and the bottom row of figures are taken 4 days later. And you can see most of the genotypes have completely filled in the wound that we made at the 4-day mark. But the risk cells, which is the first set of images on the far left, they almost haven't made any dent in their wound. It's almost the same as the day it was made. So definitely our risk cells exhibit significantly reduced migration capabilities. And you can see that quantified in the graphs below the pictures. And those graphs actually also include an additional risk donor and an additional non-risk donor, once again showing that this is a phenotype that remains consistent across donors. We also did this assay with some primary cells that we had on hand. And these primary cells, one set was homozygous non-risk, and the other was from a heterozygous donor. And we did once again see this reduced migrative ability in the heterozygous donor relative to the homozygous non-risk. And this is very interesting for us, not only because it shows that this is a phenotype that we see in primary cells, not only in iPSC-derived VSMCs, but it also shows that even a single copy of the risk haplotype is enough to induce this migration defect. And this is another phenotype that is potentially very significant for clinical purposes, because one of the things that contributes to plaque stability in coronary artery disease is a fibrous cap that forms over the atherosclerotic lesion, and that can help prevent it from breaking off into the bloodstream. And that cap is made of vascular smooth muscle cells. So speculating, theoretically, if your cells can't migrate, maybe they can't form the cap. And that's why we see reduced plaque stability. Or you could see reduced plaque stability. Okay, so we've established some pretty compelling evidence for this osteochondrogenic signature transcriptionally. We've seen that it impacts the cells functionally, it increases calcification, reduces migration abilities. But we also wanted to leverage our data set to see if maybe we could identify some genetic players that haven't been connected to vascular smooth muscle cell modulation in coronary artery disease before. So we went back to our smooth muscles or our single cell data and looked at the genes that were differentially regulated between the risk relative to the other genotypes. And we found these two candidates, LIMCH1 and CRAB-P1, which you can see are both enriched in the risk cells relative to the other genotypes. And the top two panels of the figure here, those are showing our single cell data. The bottom ones, the bar charts, those are qPCR confirmation in additional donors, showing once again that this enrichment pattern with LIMCH1 and CRAB-P1 being present in the risk cells is something that's consistent across donors. We further confirmed this at the protein level with some immunocytochemistry that you can see just there with LIMCH1 and CRAB-P1. And we also did this analysis with our single cell data to see if these two genes and SOX9, one of our osteochondrogenic markers, were occurring in these same cells. And what we found was that LIMCH1 appears to define the risk state in different combinations with CRAB-P1 and SOX9. And that's what all those different shades of purple are. The darkest one is only LIMCH1, and then the next one is LIMCH1 with SOX9, LIMCH1 with SOX9 and CRAB-P1, et cetera, et cetera. We also wanted to see how ANRIL was playing into this full equation, which, if you recall, this is the long non-coding RNA that overlaps the region, and we've previously seen that it does appear to play a role in the NB21 mechanism. Now, specifically what we saw last time, I told you all that ANRIL increased over the course of differentiation in the risk cells relative to the other genotypes. But this is actually an isoform-specific effect that we saw. So the diagram on the top right of this slide, this shows all the isoforms, well, not all the isoforms, some of the isoforms of ANRIL. ANRIL is heavily spliced. There's a lot of isoforms. But the ones in dark gray, the 4 that are marked here, those are the ones that we know show up in our iPSC-derived VSMCs. And you can see that 2 of them terminate in exon 19, 2 of them terminate in exon 13. So we in our lab tend to group them into ANRIL long, the 2 long ones that terminate in exon 19, and ANRIL shorts, the ones that terminate in exon 13. So this enrichment that we're seeing in our risk cells over the course of the differentiation, these increasing levels of ANRIL, these are specifically the short isoforms of ANRIL that are increasing. The long isoforms remained constant across the genotypes. So we wanted to overexpress these short isoforms of ANRIL to see if we could get a response from any of the osteochondrogenic features that we've seen in our data, or from the new risk gene markers that we've identified. So we did just that, induced overexpression at day 8 of the differentiation and maintained it down all the way until day 17. And sure enough, we saw increased levels of SOX9 and collagen 2A1, our 2 osteochondrogenic markers, in response to the short isoforms of ANRIL. And we also saw a response from LIMCH1 and CRAB-P1, our 2 new risk markers that we've identified from our data. So here is a lovely diagram to sum it all up, and I will run through our conclusions really quick. So we found that the 9p21 coronary artery disease risk locus promotes the transition of iPSC VSMCs toward an osteochondrogenic phenotype. We saw that the risk cells have a distinct transcriptomic signature, which includes osteochondrogenic markers like SOX9 and COL2A1, as well as 2 novel markers, LIMCH1 and CRAB-P1. We saw that the knockout of the entire haplotype reverts this signature essentially to a non-risk state, proving that there is a cause-effect relationship between the genotype and the phenotype that we see. And we saw that the short isoforms of the long non-coding RNA ANRIL are sufficient to induce the osteochondrogenic transcriptional signature, as well as our 2 novel risk markers, LIMCH1 and CRAB-P1. We also saw that iPSC VSMCs exhibit a transcriptional profile that is very similar to endogenous smooth muscle cells, showing the utility of our model as a way to study human disease, not just kind of a series of interesting things that happen, but only in vitro. And finally, we demonstrate that this whole cell state transition that we see is a cell autonomous process. It happens even in vitro, away from all the environmental signals that the cell would be that the cells would be receiving in the microenvironment of a human artery. So our next steps moving forward from this are to try to induce the risk state by overexpressing ANRIL short isoforms, LIMCH1, CRAB-P1, and SOX9 in some non-risk cells. and of course the reversed to try to knock them down to see if we can rescue the risk state. And this is something we're interested in doing both during development from IPSC to VSMC to see if we can kind of prevent them from or cause them to go to the risk state. And also we're interested in doing it with mature cells to see if we can kind of bring them back from the risk profile once they've already developed into fully mature VSMCs. And with that, I would like to thank my lab members. They contributed enormously to the work that I presented today. Could not have done it without them. Of course, my PI, Valentina, for all of her mentorship and guidance. And of course, our collaborators at the University of Virginia, Clint Miller and Jose, who is his grad student, and he was the one who did all the analysis that I showed you today. Thank you. Yes.
Question for clarify, because I have missed something, but is the difference between the long non-coding RNA on expression levels, or are there changes in the sequence of that long non-coding RNA? Do you think the difference is just that somehow in that haplotype there is misexpression of that long non-coding RNA? Or do you think that some other kind of gain of function change in the sequence?
We think it's an expression difference because we see an increase in the short isoforms. I think if we were seeing like mistaken splicing that was producing isoforms other or like short transcripts, we would also see a change in the long isoforms.
Cells versus the cells with this haplotype. There is no increase in expression of that long non-coding RNA at all in the control normal cell.
In the control, I think there might also be a slight increase over the course of the differentiation, but there's no like differences between the genotypes at all.
Yeah, I was just trying to, I'm still
Yeah.
confused about what exactly the difference is between,
Well, I think what we think is the splicing controls are being differentially regulated in the risk cells. So it's causing them to express more of particular isoforms over. Yeah. Mhm. Yes.
Testing of head genotype. So you talked about an increased risk based on the presence of one or two copies. Have you tested the head in this system to see if it's gain functioning?
Yeah, so there's another student in the lab who is focusing on a model with hemizygous knockout. So there's a donor who's heterozygous, and then she did individually knocked out either the risk allele or the non-risk allele from in cells from that donor. And she does see evidence of a dominant effect of a single allele with the heterozygote far more closely resembling the risk allele cells than the non-risk allele cells. Mhm. Yeah.
The one in isoforms, have you tried, like, what might be results, like what splicing mechanism might result in the reduction of the shorter isoform, and can you track what RBOs might be used in that process?
Yeah, so the person who's doing the hemizygous system, Shraddha, she is also looking into splicing, which we think is being misregulated in the presence of the risk haplotype. I don't know that we're looking at it like a specific splicing mechanism yet, but for sure we're looking at splicing as a kind of one of the ways that we think the region is affecting all of these transcriptomic differences. Yeah.
You mentioned you had identified 80 SMPs within this orbit. Are you looking into like any other elements that might also be playing a role in this? I know you said animals didn't completely take this focus.
Mm-hmm. Yeah, so we did not identify the SNPs. Those have been kind of previously reported. As far, you mean like how we think 9P21 is doing things and affecting stuff, or?
Yeah, I just feel like if Emerald isn't like completely overlapping this
Yeah.
focus, like, is there something else there that you think that's also playing a role?
Yeah. Well, we haven't super started to dig into that yet. I am interested though, because the region is predicted to be enhancer rich. So that could be something that is kind of causing all this.
So those short isoforms of emerald that you've seen, do they occur in other types of cells, other disease states? And what else is known about emeralds? Is it really unique to this particular system, or
Yeah,
is it out there, causing chaos all over?
Yeah. I don't actually know if the short isoforms are showing up like particularly enriched in cells from like a specific other part of the body. Um, but I know that kind of previous research into Anrol has gotten kind of mixed results. A lot of it, I think, predictably has been related to heart disease specifically because it overlaps this incredibly impactful CAD risk region. And the ways that it interacts with like CDKN2A and B, the two closest protein coding genes. To the best of my knowledge at this stage, there's a lot of kind of mixed results around it. Yeah.
That sort of suggests it's involved with something else, but not quite sure what.
Yeah, potentially.
I think that, if I can add
Yeah.
one more thing to Elsa, is there is not really a study that has done like a sort of a tissue survey of real isoform expression. So there are studies that have been described and short being upregulating add donor and long decrease, but in specific type of cells. And then if you switch type of cells, if you do blood cells, the expression is completely the opposite. So I think that there is some cell type specificity to the expression in general of the isoforms of aerial. But in our lab, we've mostly done smooth muscle, some endothelial cells. But yeah, definitely envy is not expressed everywhere. It's not expressed in IDS cells, for example, like, or in the mesodermal progenitors. But yeah, we don't know yet.
Sorry, just going back to that, it also seems like at least based on the experiments showed, it doesn't look like the normal is required normally, right? Because you show a normal donor without that region, there's no feedback, and then the affected donor have different effects, so it's clearly some gain of function.
Yes.
Region, but it's weird.
It is. Yeah.
You looked at some of the transcription factors like SOX9 to see how other cell states map onto this unique cell phenotype that you have. Have you taken kind of a more agnostic approach, just looked at the transcription regulators that were specific for your risk allele and looked at their interactions based on their ability to regulate or bind to each other to see if it's all through ANRIL, hitting each target individually, or if it's just activating a cell state network that drives it to an endpoint?
We did identify the transcription factors and that were kind of driving the transcriptional profile of the risk. We did an analysis with a tool to try to see how predict some of the regulatory relationships that you're talking about there. We didn't see a ton with that specific analysis that we did, but it also isn't something that we've continued to look into very much. So it's possible that there is still something there, but we need a different tool to see it.
Yeah, let's thank you also one more time. Extra thanks for being the first one. Yeah, they set the bar for a honey mask.
Thank you.
You're also nice job.
How are you? We hide. No, I actually now. Yeah.
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 That's cool. I've never used.
Yeah. Yeah, just terrible. Yes. Right now. These two next months, like, I have no time, but this as far as like. It's fun stuff, like. Everyone, boy, yeah, he just started. The thing what happened was that, and so then I able to have it out yesterday, not today. I don't know, every day, yes, I got it, I was like, how did it go? And then not miss the bills. Yeah. And then, yeah, nervous about it. Like, it's so bad. Then they got to like, the teacher last game. It's all little fun class.
It's a great start to.
Yeah.
Another 1 minute. I do actually get.
Okay.
It's like, well, if you just push it instead.
Okay.
So only, you know,
Yeah.
then today I walked in and.
Yeah. Yeah. You know, well, we're in Middleton in the middle schools, like 70. Oh my god. And so I just imagine,
Yeah, it's.
10-year-olds with like.
There's too much.
The girls, you look like, you know,
Yeah.
we were the adult, yeah.
It's real bad.
Yeah. This is coming, right? Oh, yeah, bro. Oh my god, yes. I'm not good. No. A stupid question about this locus that I never thought about before, and just to clarify, this is like not an inversion or anything. No, it's just a haplotype of like strong OB. Yes.
Yeah. I think, you know, like also Elsa told me that the genetics retreat, one of your students had something about the.
Oh, yeah.
Presented about, like, I was not religious, but Elsa told me that it was so because
Yeah.
you guys do the T2T.
Yeah, Joe,
Separately.
yeah, my student Joe or graduating.
Yeah.
Around, but not really around. Yeah, we've been comparing the reference, you know, and one of the things that he was saying was, basically, I know. Region that is commonly associated with a copy number error, so
Yeah.
flanked by duplication, like
Yeah.
hard to sequence region. Using the new T2T genome helps with genotyping accuracy in those flank
Yeah.
regions, and also if you're phasing variants.
Yeah.
We can get better phasing accuracy. And
I think that, you know,
including this one, which is not a copy number variant, but. still
No,
intriguing opus.
yeah, this, but still, like at one point of like, you know, when I was in my postdoc, there was another, someone at UCC that was looking at the logos and at one point it was duplicated.
Hmm.
But not only the expression in general, but
Yeah.
in the genome, the 1921 region, this region that is, you know, being took had, it
Yeah.
was just the two copies,
Okay,
like, you know, duplicated, but
yeah,
I think that there was some caveat with the system that they were using. I don't think
Yeah.
it was a true duplication. It was like, you know, their system of cloning had some fluke, but I don't think it's duplicated. But even if it's duplicated, it would be, you know, in the risk version around risk.
yeah,
So I don't think it's just one more copy that makes the trick. It's just the variants within it, whether or not they are in the risk version of this.
Sure.
Go ahead.
Yeah, this is a naive question, because I know we've done EQTL studies before, and there's certain loci that are of common inversions. And so Clemson 17 has one where you get this big block of what looks like a bunch of significant hits in one locus, but it's just because the variants called there are all exceptionally strong in the LD because they're all inverted, and it messes up
This one, I think that, the variants are technically, at least in this stretch, in very strong linkage. But then the EQTL, I think here don't really I think that for me don't make sense in terms of if you want to identify a one variant.
Yeah.
They all linkage is so,
right?
they all do what we're doing with, Carol, from my lab, I think you probably teach her some classes. Second year now.
Second year,
Yeah,
Carolina.
Okay.
Carolina.
Yeah.
So she's looking at some of these with more lines that we have from African descent individuals that do not have these linkages equilibrium.
Okay.
They don't have strong LD, so we have
Yeah.
a series of lines that have all mixed up alleles,
Yeah.
and we're trying
Is
to.
general messed up in them?
The short isoforms are not even expressed, but
Okay.
also, these locus has a strong link with CAD in a non-African population. So in the African descent population, there is no linkage.
Right.
And we don't see Eric short, which for us is a kind of a signature of disease, basically. But it's interesting because the analysis that we had in the long isoforms, it seems that they have each SNP has an additive effect on the amount of long isoforms in that. So our theory is that the long is actually protective, and the more you have, the better.
Yeah.
So
the short check.
yes, something like that. But I think that we have evidence of an additive effect of the SNPs. Yeah, which is pretty cool.
Yeah.
And we can do it basically only with those type of samples that are not in strong linkage.
Right.
So we have, lines where we have two SNPs that are in the risk version and all the other are non-risk. Or, some of the lines have 10 SNPs that are in the risk version. And they're not risk. So we're trying to use that as a, almost a perturbed seq.
Yeah. Naturally direct.
Exactly. But anyway, we'll see you around.
Yeah. Great.
Good luck.
Exciting to see that published, sure.
Yeah.
That's not your computer hooked up, is it?
No.
Okay.
Yes, she published it. I'll get a